Hello everyone, welcome to the webinar. I'm Lizzie Seguin, I'm President-elect of the British Society of Soil Science, and I'm delighted to welcome you all to our 9th webinar of the year this lunchtime. Before I welcome our presenters today, I'd like to introduce the British Society of Soil Science as hosts of today's webinar. We're an established international membership organisation and charity committed to the study of soil in its widest aspects. We bring together those working within academia and have a growing membership amongst practitioners implementing soil science in industry and those with a keen interest in soils. We're hosting 10 webinars during 2025, so keep an eye out on our website for the details of the final few of this year. Before we begin, just a few basic housekeeping. As there's so many of you here today, all your microphones have been muted. We will be taking questions during and at the end of the presentations, and we'll be monitoring these throughout the webinar. So please do ask questions. Please can you send your questions in the questions box and try and get those through to us by 12:50, just to allow us to get through as many as possible. So the webinar today has also been awarded BSSS and Neuroso CPD points. So if you're registered with either of these, please contact us directly after the event. Finally, please be aware that we're recording today's presentation. So the webinar today is organised in partnership with the Society's Corporate Member, Cranfield University. Cranfield is a postgraduate university specialising in technology and management. They work in partnership with business, academia, government and other organisations to develop and deliver applied research in innovative education. Cranfield has world-class facilities for soil research and teaching and are home to national and international soil datasets, including Landis, the World Soil Survey Archive and Catalogue, and also the National Reference Collection for Soil. So our first presenter today is Dr. Emily Guest. Emily is a research fellow in soil and plant science at Cranfield University and will speak about her work on the Nitrogen Climate Smart project, so the NCS project, which aims to increase the frequency of legumes in crop rotations and reduce the UK's greenhouse gas emissions. Her wider research interests on land management strategies that enhance agro-ecosystem sustainability at the rotation scale. So for example, the incorporation of leys and legumes. Emily is also interested in harnessing new technologies and farming systems to generate actionable insights that support decision-making for improved farm productivity and profitability. So I'll pass over to you now, Emily.
Thank you very much, Lizzie, for the introduction. Can you see my screen okay?
Yep, you're good to go.
Hey, fabulous. Hi, everyone, and thanks for coming along today. So yeah, I'm Emily Guest. I'm a research fellow in soil and plant science here at Cranfield University. And today I'm going to be talking to you about investigating the role of legumes in nitrogen climate smart agriculture through my work on the Nitrogen Climate Smart project. So I'll start off with a bit of an introduction about what the Nitrogen Climate Smart project is, its aims and its background for those who haven't heard about it before, then go on to describe my experiments at Cranfield University and the initial results that are just starting to come through from my first experiment, and then move on to the future directions that are planned for the rest of my time on the project. So first of all, the Nitrogen Climate Smart project, or the NCS project as it's known for short, is a 4-year research project which started in April 2023 and is going on until April 2027. It's got 19 partners across academia and industry and is a £5.9 million funded project by DEFRA's Farming Innovation Programme and delivered by Innovate UK. It has an overall aim of achieving a reduction of 3.4 million tonnes of carbon dioxide equivalent per year in the UK, which is equivalent to 7% of UK agricultural's total greenhouse gas emissions. And it aims to get to this figure by increasing pulse and legume cropping and arable rotations across the UK to the rotational optimum of 20%, or 1 in 5 rotations. So how do we get to that 3.4 million tonne figure? Well, legumes themselves don't require any nitrogen fertiliser because of their ability to form those symbiotic relationships with nitrogen-fixing bacteria. But they also reduce the application of additional nitrogen fertiliser indirectly as they reduce the nitrogen requirement for the crop following a legume. For example, in the RB209 nutrient management guide, which farmers use and their agronomists to decide how much nitrogen to apply to a field, it recommends that you reduce your nitrogen nitrogen fertilizer applications by 30 kilograms of N per hectare following a legume cash crop. Legumes also require, on average, less fuel to grow and harvest compared to the average cereal crop. But the biggest section which contributes to this 3.4 million tonne figure is what do we do if we do grow more peas and beans in the UK, and if we use them to replace 50% of the soybean meal in livestock diets, that contributes a massive part to the 3.4 million tonne figure. In some cases, this could be less, because it's actually found out through the early research in this project that a lot of farmers don't actually reduce the nitrogen fertilizer following a legume. So we're hoping to build more data up around to either improve confidence in reducing nitrogen applications, or in some cases maybe reduce them even more than 30 kilograms of N per hectare, depending on the environment they grow in. If you'd like to read more about the calculations behind this figure, Farm Carbon Toolkit produced a report on an overview of how to increase how an increase in homegrown pulse production and use could impact UK greenhouse gas emissions. And we very much appreciate that we can't get to that reduction of 3.4 million tonnes of greenhouse gas emissions within the scope of a 4-year research project. This is going to be a long-term initiative, and the NCS project is really set up in a way to hopefully increase impact and maximise the long-lasting impact beyond the scope of the 4-year research project. So this is the theory of change diagram, which I'm hoping to include in a paper submitting later this year, kind of using the NCS as an example project of how projects which rely on farmer engagement and behavioural change and therefore land use change should integrate farmers through on-farm trials, as well as having those more mechanistic experimental field trials, and engage farmers in peer-to-peer knowledge exchange to be able to create that impact, along with supporting the supply chain and making sure that things would make economic sense. So some early work in the NCS project looked at where we are in terms of the state of play for the UK pulse cropping frequency. This is work done by ADAS, and they reviewed several databases which are show some discrepancies in where they think we are with pulse cropping currently. But overall, it's estimated that we're currently at about the 5% mark. So quite a way to go to get to that 20%, which we need to get to that 3.4 million tonne figure. And it was determined that we have both scope for farmers who grow pulses to grow them more often, but the largest increase would be to try and get new farms to start growing pulses. So the NCS project is split up into 6 work packages. We've got people doing environment, climate, social and economic impact evaluations. We have farm case studies with farmers carrying out on-farm trials. We have experimental platforms where we have small plot studies, and this is where we sit with our glasshouse experiments. We have animal feeding trials led by MacArthur Agriculture, doing things like processing the pulses in different ways to be able to maximize the nutritional profiles of beans and see how they can be switched out and replace soybean in livestock diets. And a big part of the project is also looking at the community engagement, getting farmers involved and really maximizing that knowledge exchange. So when I started my postdoc, a lot of the field trials had already begun, and so I was reviewing what trials were already ongoing through the farmers who we call pulse pioneers, and the industry partners doing their small plot trials. So we had the pulse pioneers doing split field trials, for example, comparing a field that has half legume, half non-legume, and really tracking the crops into the following 2 rotations through ADAS's YEN network and things like soil health scorecard. And then the industry small plot trials were doing things like investigating different nitrogen rates following legumes, looking into the impact of soil-borne pathogens if we do grow more legumes more often, biocopping and different management techniques. For example, should we be removing the bean straw or not? So I was really thinking about what trials already exist and how I can best fit into the existing project. And at Cranfield, I am very lucky to have access to this amazing large glass house, which is owned by the UK Agritech Centre, and it's based at the Cranfield campus of Cranfield University. Within this glass house, we have these meter-cubed boxes of soil, which we call soil lysimeters, and it allows us to grow crops just like they would do in the field, and up to yield. We can harvest them for grain, and so we can carry out what we call a near field scale experiment, because we can grow crops that are more realistic compared to smaller pot trials, but still under relatively controlled conditions compared to in the field, where we're more at risk to the environment. For example, the really dry summer we've had here would have been detrimental to a lot of field trials this year. Each of these soil lysimeters are set on individual weighing scales, so we can individually manipulate the watering regime remotely of each different lysimeter. And we also have access ports throughout the soil, which enables us to see roots and bean nodules growing live, essentially. So we can look at things in a lot more detail than we can do in the field. And one of the big questions people ask when they see this glass house is, why is it so tall? And that's because it has this gantry-based sensor system, this box essentially that has multiple sensors and cameras inside it, and we can take things like thermal imagery and hyperspectral imagery throughout the crop's life cycle and really look at things in a lot of detail. So this is my experimental design. I had 18 of these meter-cubed soil lysimeters, and I filled half of the lysimeters with the spring bean crop and half with spring oats. So we have a leguminous crop and an alternative non-leguminous break crop that a farmer might grow if they're not growing beans in their rotation. And throughout these, I had 3 different moisture treatments, which replicate the 3 different rainfall regions of the UK. And then from all of these boxes, I followed them with a wheat crop, continuing the different moisture treatments. And throughout these 2 different rotations, I measured multiple different things, including soil analyses before and after the legumes or oats, and then after the wheat crops, for example, soil mineral nitrogen and microbial biomass C. We had those, the access ports to be able to put root cameras down and see, assess things like root biomass and length and nodulation. We had greenhouse gas collars, so we could measure nitrous oxide, carbon dioxide, methane and ammonia emissions. We had the overhead gantry system taking photographs and having so we can derive vegetation indices. We had within season and post harvest physiological measurements, including harvest index, yield, nutrient analysis, and biological nitrogen fixation through isotope analysis. We had access ports for soil moisture and also nitrate sensors kind of plugged into the soil, which can do live tracking of the temporal and spatial availability of nitrate ions at different depths into the soil. So hopefully you'll be able to kind of track how much nitrogen is fixed and how it becomes available to the following crop. So from all these different measurements, I can ask lots of different questions, but I've just narrowed it down to 2 of the ones that I was most interested in. So I am mostly interested in how biological nitrogen fixation, otherwise known as BNF, can be better estimated by using more easy to measure and quicker and cheaper ways of measuring BNF, because that can be expensive and not everyone will be able to do that. Can we better estimate it by using things like vegetation indices like NDVI? So in the glass house, we've got a gantry-based image system, but in the field you could have freely available satellite data, for example, on a much bigger scale. And can we use that information to better estimate and therefore more accurately advise how much nitrogen fertilizers to reduce by in the crop following a legume? And I'm also interested in whether greater bean yields biomass lead to greater BNF and therefore greater benefits of N to the following crop, and whether we can move to more of a system that Australia does, for example, where they use the previous bean yield to be able to better tailor nitrogen recommendations to the following crop. These are just some photos of the experiment. So you see the beans got very tall. We had to, in fact, stop them growing because they wouldn't stop. And then you can see here how we harvested the beans and the oats in the lysimeters, essentially doing what a tractor would do in the fields, cutting them off at 10 centimeters height and leaving a stubble. And then the wheat, we essentially direct drilled with our hands into the soil, the wheat seeds, and we got a really good wheat crop off of that. And we've just sent the ears off for threshing, so hopefully we'll get the yield data back very soon. These are some images of the root imagery that we got from the bean lysimeters. So here you can see the nodules in situ inside the boxes. And from that, we can use programs to derive the number of nodules that and roots, the volume, length, width, and area. And when we cut open these nodules, this was a sample that I took when it was time to take the samples for BNF analysis, and the nodules were pink. So we're very excited that we did get some biological nitrogen fixation. So these are results from the bean, oats in yields and biomass results. So we tended to get a greater yield and biomass in the lower water treatment, which was initially a surprise, but after looking more in depth at the rooting imagery, it seems like we did get some root and nodule death at the higher water treatments. So we think that actually the low water treatment is essentially ideal for the crops, and then it got too wet for the crops in the higher water treatments. But what the most important thing is that we've got basically a gradient of yields for both the beans and the oats, and we can use that to be able to look at correlations between BNF and physiological parameters. We did also see a correlation between more yield and more biomass, so it'll be really interesting to dive down whether that means also greater below ground volume, and therefore does that lead to also greater nitrogen contributions from the below ground biomass. These are the results from the SMN, soil mineral nitrogen analysis. So we did get, so time point 1 is before the legumes or the oats, time point 2 is after the legumes or the oats, and time point 3 is after the wheat crops. So we did get either a maintenance or an increase of available N overall throughout the 2 soil profiles that we measured, suggesting that we did get BNF in the beans. And we see a build-up particularly of available N after the legume in the upper soil profiles, which is what some of the partners saw in their small plot trials. So that's reassuring. And then obviously it goes down after the wheat crop's been grown. And then looking at the bean nodulation, we do seem to see a similar trend to what we saw in the bean yields and biomass results, where we get greater nodulation at lower water treatments in line with high yields and biomass. Although this wasn't significant, it seems to be a trend towards that. But we did see how the higher water treatments had lower nodulation volume in their area at lower depths. This is a little video showing the gantry image system in use. So the first question was, what growth stages of the beans do we target to be able to relate vegetation indices to BNF, hopefully, because this hasn't been done before to our knowledge. So we selected 3 different growth stages across the bean growing season and used all of the cameras available. So we've got hyperspectral, RGB and thermal imagery. Here you can see what the images look like just from the RGB imagery. We chose growth stage 19, which is quite early on in the bean growing season, where we've got 9 or more leaf pairs unfolded. Growth stage 55, when we start to see the first flower buds. And growth stage 63, where we just start to see the first flowers forming. And this is currently still being processed, but here you can see an idea of how we derive vegetation indices from the hyperspectral imagery that's been taken. So we'll essentially take a square of the image inside of each of the boxes, and from this we'll get a numerical value for each of the boxes, which we can then do stats on and relate it to things like BNF. This is some initial results from the wheats, because just sent off the ears for threshing, but this is really exciting because we are seeing that roots have significantly more length, surface area and volume after a bean crop compared to an oats. So we've got here the high, medium and low water treatments. You can see that overall the in every single water treatment, this is the root length, but the volume, surface area, and a number of the roots all follow the same trends. And we're particularly seeing that after beans, roots are developing a much greater surface area and volume and depth. So it's starting to raise the questions of, are they following the same root channels that the beans are forming through their tap roots, which oats don't provide for the soil? And it enables us to ask questions like, could this help we access water and nutrients better, leading to potentially improved yields and greater resilience against droughts like we've had this summer? So my next steps with this experiment are to focus on the wheat post-harvest analysis. So we'll have the harvest index, nitrogen harvest index, and yield when we've got to get all the threshed wheat back. And I've got lots of greenhouse gas and nitrate blade data to process. And then I'll start looking at investigating associations using correlation analysis. And also what I want to do is quantify the exact moisture of the soil at key growth stages, because maybe it is at some point that because we diverge the water treatments, maybe it'll be interesting to quantify the exact moisture at different growth stages and see what impact that had at certain times, which are key to wheat yield formation. So over this winter, I'm going to be analysing a lot of data and writing some papers, but also thinking about the next experiment, which are going to be in the controlled growth rooms that we have at Cranfield University. So I'm going to be testing the question, do soils with legumes integrated into crop rotations have greater resilience to climate change pressures? So I'm planning to, what I'm doing is trying to find a farm that has some soils that I can use and take back to the university and test whether soils with legumes, so a reduced response in greenhouse gas emissions under climate warming and drought, and do legumes provide soil health benefits, which is able to mitigate things like nitrate leaching and enhance resilience. So this is my general experimental design for my next trial, which will be to get a variety of soils. So one that has a non-legume background, has never had legumes grown in the past, and legumes at different intensities. So one that has just had one legume grown, and then one that's had a really strong history of legumes for the past 20 years or so. And going into the field, taking in-field measurements to look at the physical differences that growing legumes causes, for example, on water holder capacity and soil aggregate stability, and then taking them back and putting them under climate change pressures like drought, so causing drought at key growth stages, which are key to wheat yield formation, and then also testing resilience metrics, like what does this do to soil biology, yield, do soils with legumes show a reduced nitrate leaching or greenhouse gas fluctuations. Before I end my presentation, I just want, if any of you want to hear more about the NCS project and are particularly interested in reducing our reliance on soya in the UK, we have a related conference to the NCS project, which is called From Soil Sustainability, which is next January in Peterborough. Thank you all for listening, and thank you to my supervisors at Cranfield University in Nottingham, and also the wider NCS project team who have been really helpful in giving their advice. Thank you.
That was great. Thank you, Emily. So just a reminder, if you've got any questions for Emily, please pop them in the question box and we'll take them at the end of our second presentation. I would now like to introduce you to our second presenter, Dr. Sam Hibdige. Sam is a research fellow in Soil Microbial Metagenomics at Cranfield Uni and will be speaking to us on changes in the soil microbiome during restoration. Sam is a bioinformatician who is interested in changes of structure and function of soil microbial communities. He's currently focused on analysing amplicon sequences between restoration sites of different land use practices to ensure resilient restored ecosystems. It sounds fascinating. So over to you, Sam.
Wonderful, thank you. So yes, hello, I'm Sam Hibdige, and I'll be talking to you about some of the outputs from the Restrico project, which is focused on restoring resilient ecosystems. So Restrico overall is quite a big, ambitious project that looks at everything from trees, plants, invertebrates, small mammals, down to soil. And I'll be talking to you about the soil section, as that's the bit that I was working on I was working on the metagenomics side, so all of my stuff was computational analysis. So I wasn't actually allowed to go outside and see all these wonderful sites most of the time. And so Cranfield was responsible for most of the soil work. So alongside the metagenomics I was doing, there was a lot of chemical-based stuff as well. So pH, pure organic carbon that was done in the labs here. So backdrop to the Restreco project, as I mentioned before, it was to try and look at this ecosystem as a whole from a bunch of different metrics to try and link complexity and resilience. Now, I think they're normally presumed that a complex ecosystem is resilient, but I think it's one of those things that isn't often tested or proven in this context. So the idea was to take a bunch of measures from biodiversity, multifunctionality in different stages of transition from a degraded state and identify key determinants of complexity and try and link it to this resilience. The idea being that resilience is probably now something we need to be aiming for as opposed to just complexity. As with changing climate, something that was once viable might no longer be in the future. So a lot of the methods are the same or similar, but the goal is a bit different and more flexible. So Jim Harris, the lead on the project at Cranfield, likes this analogy for resilient ecosystems, that a resilient ecosystem is this off-road car on the left, where every time it hits a bump or perturbation, the trajectory is unchanged and it forgets the previous perturbations, which in an ecosystem would be flooding or drought or disturbance. And this is what we want to aim for. We want something robust that you don't have to constantly put money into to restore it to a state. Whilst a low resilience one is Buster Keaton on the right, hits the bump, everything collapses to a new stable state, but it's less complex, it provides less ecosystem services and is less useful. So how are ecosystems currently assessed? A lot of the ways restoration has been done is people going, there used to be this here, wouldn't it be nice if this was here again? And so plans are drawn up to try and build an ecosystem to allow this species to exist in this area once more, and that often comes with native species and all the wildlife that comes with it. But the facetious example that I use is that when they were digging the foundations for Nelson's Column at Trafalgar Square, they found hippo bones from the last interglacial, and you would look at me as if I was mad to say there was hippo on the Thames, we should reintroduce it. So it's a bit more nuanced now. I think the idea is that we shouldn't just be using these historic lists, but putting it in the context of what will happen and how do you make something resilient as opposed to how it was when it last disappeared. Again, a lot of the methods and goals are going to be the same. It's just trying to do it with a forward-looking view as well now. So the Restreco project itself is looking at 2 restored habitats in the UK, these broad leaf/mixed woodlands and these short grasslands, as these are the 2 most restored habitats. So in the woodlands, we had sites in England and Scotland, whilst in the grasslands, they were only concentrated in the south of England. That's not because we were fed up of going up and down to Scotland, it's because I think there's only one major chalk grassland in Scotland, so they had to be concentrated down in the south. So this presentation is going to be focused on the grassland results, whilst previously I've spoken about woodland stuff, and I will reference both as I go. So just to give you a very quick overview of the project, I know it's not soils, but yeah, we've got the invertebrates, we've got predation, which is that weird little green Play-Doh thing that has teeth marks in that you can use as a proxy for what's attacking what. We have the plants from the canopy, the small plants, pollinators, how dense the woodlands are themselves. We have birds, we have bats, and we have these soundscapes, which has produced a large amount of data, which I believe is still being analysed. And then, of course, the most important stuff, the soil, looking at bacteria and the fungal communities, as well as the associated biochemical metrics that go with it. So, as Lizzie mentioned, I have largely been involved in amplicon sequencing, which is a bit cheaper, a bit easy to do than the more expensive shotgun sequencing, which looks at everything. So amplicon looks at a small conserved region that is present in your host, and you can sequence this much smaller region rather than the whole thing, and it's quicker and it's cheaper. And so the example for the bacteria here is this 16S gene, which has a bunch of variable regions dispersed amongst the length of the gene. And the idea is that, yeah, it's conserved enough that it's present in all of the organisms you want to look at, but it's different enough that you can tell them apart. And generally for amplicon sequencing, you can get resolution to about genus level. So traditionally, you didn't even have to use the whole gene. You could use sections depending on what you're interested in. So for the Restrico project, we used this region here, which was previously established by this Earth Microbiome project, as we weren't interested in developing something new on that side. It was using these metrics to feed into the larger information overall. And the analogy I use for this is car badges. If you wanted to get a good idea about what sort of car you are, you'd go to your conserved region, i.e. the middle of the bonnet at the front, and you would have a enough variation to be able to tell the models apart to genus level or make, and you'd be able to make some assumptions based on the genus alone. So the grassland sites before they were restored were all previously agriculture. The woodlands had a bit more variation in their former land use, but it was a bit more clear cut with the grasslands, and they have all been restored within the last 7 to 76 years, with a few older examples to look at. So quite a bit younger than some of the woodlands, but still enough time to begin to get some data. And the many metrics that we measured, main ones I'll be talking about in this presentation, is the establishment method. So whether they were left to naturally regenerate or whether they had a bit of help in terms of undisclosed seed mix or addition of green hay seed to help them along. They also had varying management strategies, normally involving some form of livestock, either sheep or cows or a mix, although a few did cutting as opposed to grazing. So on to some of the results. First, we're going to be looking at alpha diversity, and alpha diversity is useful metrics for looking at variability within sites, and it gives you a bit of a snapshot of diversity, evenness, and can give you an estimate of things you've also missed. So here for Shannon diversity, all of the 3 establishment methods, green hay, natural regen, and some sort of seed mix, performed quite well on the Shannon scale, but natural regeneration, interestingly, was significantly lower than either that used the seed mix, but all is still quite high. However, looking at another alpha diversity metric, FAIF, FAIF takes into account phylogenetic relatedness as well. So you score lower on the scale if you've got more species that are closely related than as opposed to a diverse mix of less closely related species, is where natural regen seems to score slightly higher. So it might have less diversity compared to green hay and seed mix in terms of number of species or genuses, but when you take into account a wider net, it seems that it comes out on top, which was quite interesting. Moving on to the beta diversity. Beta diversity looks at differences between samples, whilst alpha diversity looks at variation within samples. And this produces this lovely looking 3D principal coordinate analysis. It's effectively a matrix of differences, and there's a couple of ways of calculating it. The ones I'll be showing you are these unweighted unifracts, which show the effect, which are more weighted towards rare taxa. And this is also a phylogenetically informed metric, so it's taken into account the relatedness of the species as well. And so at this point, the plots I'm showing you have all of the repeats on there, because 5 samples were taken per site. So you do get a bit of a mess of clustering on this one. With the, this is just showing you the bacteria. I did do the same with fungi with the ITS region, which was a bit more clear cut. But you are seeing significant differences between the groups. And so there do seem to be a couple of clusters based on establishment that are significantly different from the other ones. I normally have a bit of a spinning graph so you can appreciate the 3D nature more, as there are certain angles where one group will appear separate from the others, but I didn't have time to include it in this, sadly. But it did seem like there was a significant difference still in the establishment method, but I need to follow up with the statistics to take into account the age as well to make sure that is still consistent, and whether you get a larger effect on age on these clusters based on establishment, or whether they begin to form together based on that. These are beta diversity based on pH, which is nice to see. It is well established that you get different bacterial communities based on the soil pH. Because these are chalk grasslands compared to the woodlands, we didn't have such a range of pH, but you still see a clear effect of different communities forming under lower pH than high pH within these chalk grasslands. The effect of age was a bit harder to pick apart. One thing that I forgot to mention actually is that the percentage variation explained by these axes is still incredibly small. In the woodlands it was low, but in the grasslands it seems that the percentage variation being explained by all of this is still quite small. So it seems they are more similar than they are different. There is small variations caused by various factors that we are able to detect, and even if they're statistically significant, they are accounting for a small amount of the variation. Nevertheless, when you go to axis 3, 4, and 5 for age, you do see a gradient from low in pink to over 100. We had a couple of 100-year sites for grasslands on the right here, and you do get a gradient that you can detect on these beta diversity analysis. And there we go, highlighting the fact that we do actually have incredibly slow and small percentage composition in these later axes. So this is a quick overview of the effects of the fungi. Similar patterns were observed. You did get a nicer separation between the natural regenerated sites versus the green hay and the seed mix, sort of further building on the stuff we saw in the alpha diversity. You get the same effect of pH, where you get different fungal communities in lower pH than high pH, relatively speaking. And there was, an observable effect of age in the later axes of the principal coordinate analysis. Moving on to some of the taxonomy data. So some of the latest stuff here was done by some of the group project students on the bioinformatics course here at Cranfield. Sort of gave them the data to play with, and they came out with some quite interesting functional data, which I will be showing to you now. I've put their names in the bottom right, so big thanks to all of them. So with the here is taxonomic data. You take the sequenced amplicons, you compare it to a known database, and you can assign it to phylum, class, order, genus level depending. This is data shown to you at the phylum level, and it's also been collapsed to site level at this point. So you've no longer got the 5 repeats. It's just one site is indicated per bar. And again, you've got this, the natural regen is once again standing out as different from the other green hay and seed mix, where you've got this higher percentage of Verrucomycrobia. I always tend to say that wrong. It's one of those ones that apparently they live in association with eukaryotic hosts in ectosymbionts of protists and nematodes, but it's something that sort of need to speak to an ecologist do and ask them why this might be in soils and in particular in grasslands, and what the significance this has for the soils and the restoration. So same was done with the fungi, although with different database. And again, natural regen is looking different by eye to the green haybus and seed mix, but I need to put some stats to it and look at the overall trends. The next slide here is picking out different species clades that are different across different establishment methods. In this case, this allows you to basically transform the data into a slightly more linear fashion. So you can do this analysis and pull out significant taxonomic groups, taxons, on the right. So on the y-axis, you've got this W statistic. Basically, the higher W statistic, the more models it was where the taxon was differentially abundant with regards to what you're interested in. So here, this pulled out a couple of statistically significant taxons, this Blastococcus and Sporangiaceae, which I apologize if I butchered the pronunciation there. They were both more abundant in the green haybush established sites and the seed mix plots versus the natural regeneration. And both are actinobacteria that seem to be resilient under disturbance and favoured these established sites of the natural regeneration ones. We have the same in the fungi, except this time we have this Gibellolopsis and this Metorhizium, which came out as statistically significant. The Gibellolopsis is apparently known plant pathogen, which was not present at all in the natural regeneration sites, but was higher in the green hay sites and a bit more moderate in the seed mix sites. So don't know whether this is just an outbreak or whether these natural regeneration sites are better at protecting against these plant pathogens, but that's something that needs to be dug into a bit deeper. And the Metorhizium, I've put it up here on the right, that's actually a cockroach infected by this fungi. This was highest in the natural regen, so swings and roundabouts, and negligible in the green haybush and the seed mixes. So yes, these are volcano plots. Normally you get a negative side as well, where it looks like a volcano erupting and you're able to see stuff that was less prevalent in others. Apparently this was because the establishment methods had more than 2 comparisons, and therefore it only outputted the highest stuff. So that's something I'll also need to be looking into later as I'm writing this up. And then finally, with the amplicon sequencing, you can do some forms of functional analysis where you can do inferred functionality. It is not as strong as the functionality you get from shotgun sequencing, but it allows us to get a bit of a snapshot. So this is just for some of the fungi, where again, natural regeneration is standing out by these undefined saprotrophs, sadly. Overall, a lot of the guilds that we were seeing were these saprotroph guilds across the grasslands. And for the bacteria, we used PyCrust, which gives you a lot more pathways that are up and down regulated. So it's quite a huge data set to sort of go through and pick out interesting pathways, why they might be up for regulated in some and down to the others. So yes, big thank you to all of the Restrico team that have been involved in this, especially Soils at Cranfield, and for the Digging Deeper group project, which dug a bit deeper into the functional analysis. So there are quite a few papers coming out from this project. One at the overall stuff about how all of this feeds into an SEM, there with Emily Waddle as the leader from that, so look out for that. There's a very interesting remote imaging one coming from Will Rust, which should be out soon. There's all sorts of insect specific, spider specific ones that should be coming out, and I'll be working on some soil specific ones as well. So yes, thank you for listening.
Thank you, Sam. That was really interesting, really fascinating. So we've now come to the question and answer section of the webinar. So just a reminder, if you have any questions for either of our speakers, please do put them in the chat. We've got a few coming through, so I'm going to start with you, Emily. So we've got a question about you'd compared legume and non-legume crops in your experiments, and just asking about whether you did or whether you would have liked to have integrated a comparison with and without fertilizer there.
Yeah, so I think I kind of messaged back, but I can extrapolate it a bit more by talking, which is easier, isn't it? Yeah, so I think that would be a really interesting question to compare biologically fixed nitrogen compared to the applied nitrogen through the normal fertilizer means. I think the first step though would be what we've done here, which is comparing a legume versus non-legume, because I guess if you were doing that different types of nitrogen experiment, you'd have to have a non-leguminous crop that you'd apply the artificial nitrogen to, because you wouldn't apply that to the legume. But yeah, I think that'd be a really interesting next experiment, and maybe once we've got hopefully a way to better estimate how much nitrogen has been biologically fixed, then we can kind of do I guess an experiment alongside where you add that same amount of artificial nitrogen and see what happens there with greenhouse gas emissions. I think particularly interesting in how that becomes available to the following crop, I think, because I think there's some questions about does the biologically fixed nitrogen become available to the following one, or is it actually locked up until the one after that? So yeah, I think that'd be really interesting to explore further.
There's always lots of other treatments you'd like to include in experiments.
Oh yeah, there's so much more I wanted to do, but yeah, I was basically fixed to these maximum of the 18 boxes, so I'd love to have done different soil types as well, and yeah, you have to kind of constrain it a little bit, otherwise I wouldn't have enough hours in the day.
Yeah. No, thank you. We also had a question about the crop sensing, which I was quite interested in, sort of the potential of that. So someone's asked about, I know you've commented in the chat, but I think it'd be interesting for others about whether you were planning to use satellite NDVI analysis as part of the trials, whether that was included elsewhere in the project.
Yeah, definitely. I think so. I'm not doing field trials myself. I've only got the glasshouse experiment and the controlled environment experiment, but we have got a PhD student at James Hutton Institute who's got some trials with beans, and so he's looking at using drones, but I guess we could also use satellite data at the same time, and he's picking out the same growth stages that I am in the glasshouse, and so if we do see a correlation between BNF and the vegetation indices in the glasshouse, it'd be really interesting to see if that also applies at a field scale, whether we do get enough kind of detail of the imagery at satellite, or whether it only works at a drone scale. Yeah, so that's hopefully something we're going to be looking into and producing a paper onto, so I'll watch this space.
No, definitely that'll be interesting, because measuring biological nitrogen fixation is quite challenging, isn't it? So if we could have a way of measuring that with sensors, that would be a significant step forwards.
Yeah, because I'm not sure if people on this call know, but to measure actual biological nitrogen fixation, you need to take a few plants that fix nitrogen, but also similar non-fixing crops in the same field. So I use the oats as a proxy, and then you have to grind them all up and then put them in these tiny little tin cubes, and then we'll send them off to America for stable isotope analysis, and it's quite expensive and definitely something that a farmer's not going to be doing. So it'd be really good if we had a quicker and cheaper way to be able to estimate this.
So you mentioned that sort of in-depth way of having to determine biological nitrogen fixation. Is that have you got that comparison as part of the project anywhere? Is anyone actually doing that measurement?
Not in the field trials, I don't think, but yeah, potentially it's something I could suggest, because I know there are some that are flying drones and obviously have satellites flying over as well. So yeah, potentially that could be a little add-on project that could be interesting to have some more replicates throughout the partner trials in the UK as well.
Okay, thank you. Just one final question for you before we come to Sam. This is for me, because I noted that you'd said about you'd got greater nodulation at the lower water treatment, which was interesting. I was just wondering why that was, and whether you thought whether you had some waterlogging in the experiment that was influencing the nodulation. Yeah,
so in the imagery we did see some nodule and root depth at the higher water treatment. So yeah, I think that's what was causing that, and I think it's interesting to see that with higher nodulation we get that higher bean yield and biomass. So it's interesting. It'll be really interesting to get this biological nitrogen fixation results back and see whether they were actually more active when there was more of them as well. So yeah, I think there was waterlogging at the higher treatments, which wasn't what we were aiming for. We're hoping that the medium would be the ideal water treatment, and then we'd have more of a drought, and then more of a higher water treatment. But we've still got that gradient in the yields and biomass results. So hopefully we'll still be able to do this big correlation analysis and delve more into that.
Okay, no, thank you very much. Sam, we'll come over to you now. We've got a question in the chat from Lexi, who said, how could you explain why there was apparent contradictory effect of the establishment method to Shannon and Faith measures of alpha diversity? And so there's a couple of parts of this question as well. And overall, what's your opinion on whether natural regeneration is better or worse than seed mixes for soil restoration? And can it be detected by the relatively short-term experiments that we do? So quite a complicated question there.
Yeah. So with the alpha diversity metrics, they're generally based on how many species they are and how even they are. And a lot of them weight them in different ways depending on what you're interested in. So Shannon is one of the classic ones that does just that. It looks at the number of species and how even they are. But Faith takes into account how related they are as well. So this instance for establishment, it seems as if the ones that use the green hay or the seed mixes either had more species and were more even than natural regeneration. But then when you're looking at how distantly related they are and what sort of breadth you are getting in the natural regen, it seemed to be higher. It's just two different ways of looking at the diversity you've got within the sample. As to which one is better, that's very hard to say, because it a lot depends on what functions though that breadth of species is being brought in the natural regeneration sites. In some of the later work packages, work package 3 was looking at drought. So that looks at a couple of these different sites with different establishment methods and how the soil microbiome and plants and everything else fares when you drought them for a long period of time. So some answers could potentially come from that. And there was also some intervention sites as well, which are still waiting to be processed with the shotgun sequencing, which look at adding sugar or carbohydrates to the grasslands as well to see whether they can be rescued in terms of the stress as well. So it would be interesting to see whether or not natural regeneration fares better in any of those later on. And then as to short-term experiments, it's tricky because I think Restrico did a very good job at capturing a wide breadth of stuff, but then as a result you lost a bit of resolution when it came to actually digging down into real specific details. And it would be very interesting if after X number of years, it didn't matter how they were established. After this amount of time, they became similar anyway. I think a lot of the grasslands are fairly newly restored, so there is a weight towards grasslands that were 20 years old or so, which in itself is still interesting that there's still a signal you can detect from how they were established. Yeah. And then there's quite a lot of other stuff that we have now. I think we've got fungi and bacterial biomass that can be added into the sample as well. So into the analysis as well, so we can see how much was actually in there and whether you've got any significant changes in the volume of stuff that was there. And there's all sorts of ecological metrics as well that I need to have a play with and see anything significant comes out.
Excellent, thank you. It highlights the value of the value and importance of long-term monitoring, doesn't it? And it's something that we've talked about on webinars in the past. It sounds like you've got enough to keep you busy for quite a bit longer as well.
Yes, well, especially since I'm no longer working on Restrico, so I need to finish it off in my spare time.
I was wanting to ask whether you were seeing any evidence that there were particular indicator species that could potentially indicate a successful restoration.
Yeah, well, I think the hope of the project was that if we boil all of the metrics down from the plants, invertebrates, soil, etc., we can do one metric to rule them all, as Jim said, and if you measure this, this, and this, you can tell whether it's a healthy, resilient ecosystem. And sadly, well, I guess it's two sides. It turns out that for the SEM, there wasn't any overriding things that you could just plug in. All of them are quite important to be measured. So it sort of just, highlights the importance of monitoring, and you can't really just look at one and infer everything else. I will say though, in the SEM, the soils did come out as some of the main drivers, so I was very happy about that.
Okay, so it might be a similar answer to our final question, which was whether you think there's potential for us to use these sorts of approaches and methods to assess the success of a restoration.
Yes, yeah. I think that was the idea. I think because a lot of it was coming in the later work packages in terms of tying that in with functionality and resilience. I think that is the goal, and to try and frame it differently than we have before. I think, the idea is to be able to not only assess but monitor this potential sort of breakdown in ecosystems before it happens and whether it can be intervened. So yes, I'm very, very hopeful that some interesting stuff does come out of it, and some of the papers are really quite cool.
Okay, brilliant. Thank you both for two really fascinating presentations. That sort of draws our Q&A to an end. So on behalf of the Society, the British Society of Soil Science, I'd like to express our thanks to both of you, to both Emily and Sam, for coming along to present today, and the support from our Society corporate partner, Cranfield University. Thank you all also for attending, for joining us this lunchtime. You will find a quick feedback survey when you leave the webinar, which would be great if you could just take a couple of minutes to complete. The recording of the webinar will also be available after the event on our YouTube channel, along with all of the other Zoom into Soil webinars. Our next webinar is planned for Wednesday, the 8th of October, so keep an eye on the website and follow up emails for more details. So we would just like to bring your attention to our upcoming Early Careers Conference, specifically for BSQ, British Society of Soil Science members, on Monday the 1st and Tuesday the 2nd of December. So a wide range of early career members from academia, industry and policy will come together for a variety of theme sessions, including talks, posters and workshops in Manchester to hear and to learn about the latest research and developments within soil science. It's free for the Society's early career members, so now is a great opportunity to come on board and join us as a member if you aren't already. So I hope to see you all at future events, and in the meantime, thank you again and goodbye.